Okay, so be aware of a couple of optional point opportunities. Actually, there will be 2 more next week on, I believe, Monday and Wednesday also. I'll get the dates for you after class. We have 3 physics candidates coming in for our physics position next year. And we like to have students come to the teaching demonstration. And one of the things we do during the teaching demonstration is we have some kind of evaluation forms. You can give us some input about how you thought they did. Many of you will be taking physics either next year or the year after. And so if you would like some input into who we hire, try to come to these meetings. We'll have one tomorrow at 3:30 to 4:30. Dr. Williams will be speaking from 4 to 5 about Ebola. So both of those are worth 25 optional points. Okay?
What room is the physics?
Physics, I believe, did I say 308? And I think it's 308 in the email, yeah. And Ebola will be in here. Okay? I just talked to Austin, and he said he put together kind of a Jeopardy game for you for review for the exam if you come to the mentoring session tonight. And so you should be hitting your studying hard now. On Monday for the exam, we are bounded kind of by classes before this class and after this class. And so I will let you start as soon as you can come in when Dr. Rhodes' class ends on Monday. And then you've got to finish by 5 till, because he's got a class the next hour. So time yourself accordingly. If you want to look at the old exams to get an idea of length of exams, I'll tell you tomorrow when we do our review how many questions there will be and at what type. Okay. So come prepared to class tomorrow for review. Okay? So I kind of rushed through something that I want to go back and spend just a little bit more time on, talking about the history of membrane models and how they've evolved over time, getting us to the point where we have the most currently accepted model, which is the fluid mosaic model. So we talked about the fact that the early concepts of membrane models were pretty simplistic. Back before the turn of the 18th century, we believed that membranes contained lipids based upon observations about like dissolving like, nonpolar substances crossing membranes more easily than polar substances. Part of that was actually the result of observations about what kind of substances were good for anesthetizing people. In order for something to anesthetize someone, it's got to get across something we call the blood-brain barrier, and it's got to be able to cross membranes to do that. And they found out that compounds like chloroform, which were hydrophobic, were much more effective as anesthetics than polar substances were. And so these observations came from a number of different sources. But the concept that membranes contain lipids was pretty fundamental. And then shortly after that, the idea was modified a little bit that the membranes weren't just any types of lipids. They were these empathic phospholipids. So the idea that the phospholipid had both a hydrophobic and hydrophilic portion became an important concept. That created some problems for envisioning how a cell membrane might be constructed. And one of the earlier concepts by Langmuir, who is credited with this idea that the membrane was made out of phospholipids, is that phospholipids could be happy or stable in an aqueous environment in this particular type of arrangement. And so if you had phospholipids floating on the surface of water, that they would orient themselves so that the polar or hydrophilic heads would associate with the water. And then these hydrophobic tails, these fatty acid molecules, would then be kind of sticking up in the air. And we could form a filamentous layer on top of water. The problem with that is that cells exist in aqueous environments, and this isn't air out here. Okay? Typically cells are bathed in fluids. And so we've got the cytoplasm down here, which has a lot of water. and we have the environment, which has a lot of water. And these particular type of arrangement wouldn't work if this was water up here, because these hydrophobic tails wouldn't want to associate with that water. They would be unhappy in that particular arrangement. So a few years later, in 1925, Gorder and Grindle did an interesting experiment. They worked with red blood cells, and they actually measured the lipid content of red blood cells and quantitated how much phospholipid there was. And then they also measured the surface area of the red blood cell. And what they found was there was twice as much phospholipid as there was surface area. And as they thought about that a little bit, they said, well, that could be explained in a couple of ways. The most attractive to them was the fact that this suggested a bilayer for a membrane as opposed to a monolayer. And the nice thing about the bilayer is it gave you a structure which was stable in an aqueous environment. Okay, so if this is your water environment, this is the cytoplasm, this phospholipid bilayer was very happy in that particular type of environment because these hydrophobic tails were separated or sequestered into the core of that particular membrane where water was excluded. In 1935, Dabson and Donnelly said membranes are more than just phospholipid bilayers. Proteins are an important constituent of the membrane. What Dabson and Donnelly really didn't appreciate, though, was the role that proteins played in membrane structure. They thought they were pretty much just kind of structural molecules, and they really didn't have an insight into the important functions that these particular proteins played. They envisioned a phospholipid bilayer surrounded by sheets of protein, both on the environmental face and on the cytoplasmic face. So that particular sandwich molecule that I referred to, or that trilaminate structure that Robertson saw with his electron micrographs in the 1950s, suggested this arrangement of a sandwich structure with your layer of proteins on the outside, layer of the proteins on the inside, and then your phospholipid bilayer sandwiched inside. Well, fast forward to 1975 and the fluid mosaic model. And the fluid mosaic model really did explain a lot of the observations that were becoming inconsistent with the Dabson and Donnelly model. And the other important thing about the fluid mosaic model was that it really led to a greater understanding of the functional role that proteins play within the membrane. They weren't just structural components, they had important functions. So, here's the fluid mosaic model. Still have our phospholipid bilayer, but inside that phospholipid bilayer there are some other lipid type of compounds. You recognize these? These little 4-ring, 5-ring structures. What are those? Yeah, they're steroid molecules and specific cholesterol. Okay? So the fluid mosaic model incorporated this idea that, well, yes, the phospholipid bilayer is very important. It's not the only game in town, that there are these cholesterol molecules which are important functional parts of the membrane. And then we've got all these proteins, and they are not just sandwiched on the surface, both cytoplasmic and environmental, but they can actually span the membrane, and that's going to provide some important function for that particular membrane. Those proteins that span the membrane are called transmembrane proteins. And one of the things that transmembrane proteins are often involved in is transport, moving substances across that membrane. Remember, we need transport proteins in both active transport and facilitated diffusion. Some of the proteins were pretty much confined to the outer edges of the membrane, both cytoplasmic and environmental. Those that are found on the outside or the edges of the membrane are called peripheral proteins. Some proteins can be embedded within the phospholipid bilayer. Those are called integral proteins. So they aren't just in one location. They're not just these flat sheets. They're interspersed all throughout the phospholipid bilayer. That's where the term mosaic comes from. And more importantly, those particular proteins have the potential to move throughout the bilayer. Now, sometimes their movement is restricted, particularly if those proteins are associated with elements of the cytoskeleton. So that cytoskeleton can offer kind of anchorage point for some proteins so that their position within the membrane maintains a relatively fixed position. Others are kind of floating around like icebergs in an ocean. It wasn't just the proteins that moved, however. There were also observations that were made that suggested that the lipid molecules themselves could move throughout the bilayer. And that was an important observation in the understanding that the membrane was a fluid structure where components were constantly interchanging with one another as opposed to a static structure. Now, there are some restrictions as to what kind of movement is tolerated in terms of phospholipids within the bilayer. Okay? One type of movement that is not very tolerated well at all is a movement called flip-flop. And in flip-flop, this phospholipid would transfer from this edge of the membrane, the one facing the environment if you like, and flip over here so it was on the other side of the membrane, the cytoplasmic side. Why would flip-flop be relatively rare? I would tend to argue against that not happening. Okay? Well, that's good possibilities that these tails could now be exposed to water. What's the other possibility? That these heads would have to pass through this hydrophobic core, and they wouldn't be happy there either. However, within one half of the bilayer, for example, either this half or this half, phospholipids can move laterally from one position to another with pretty great ease. And they actually did some measurements, and they measured that a phospholipid molecule could move at a rate of about 2 micrometers per second within its half of the bilayer. Now, that's probably hard to contextualize and say, what does that mean? Is that very fast? So let's say this represents a bacterial cell, like an E. coli. It's not uncommon for E. coli to have a length of somewhere between about 2 and 4 micrometers in length. So what does that tell you? If I have a phospholipid molecule in this part of the membrane, down here at this end, in 1 second, that could have migrated almost to the other end. That gives you an idea of the dynamic nature of that particular cell membrane and how readily things can move. Think about this analogy. Think you're in a very crowded room, and you're sitting at the back, okay, and it's full of people. You want to get out of the room, okay? Everybody can kind of be milling around and in fairly fixed position, but you might be elbowing your way through that crowd. So you're like a phospholipid molecule moving within that structure, which has some definition, but individual molecules can change position within that particular structure relatively easily. Whoops. So what does it mean when we say a membrane is fluid? It means that the components that make up the membrane aren't in fixed position. Proteins can change position within the phospholipid bilayer. The molecules that make up the bilayer can change position. And so this gives this particular membrane a dynamic nature as opposed to a static nature. The membrane's not just like the piece of cellophane dialysis tubing you're using in lab. It is a much more dynamic unit than that is. Why is it important for a membrane to stay fluid? What would be the consequences if a membrane lost its fluidity. Yep.
Be able to form endometriosis.
Yeah, it couldn't do endocytosis. It couldn't pinch in and form those sacs. It couldn't do exocytosis because vesicles could not fuse with it. The endomembrane system could not form vesicles that pinched off of the ER and went to the Golgi apparatus, allowing you to carry proteins into the Golgi for further modification. So that particular part of membrane function would be compromised. There's something else that would be compromised if you lose fluidity. And that is, if a membrane loses fluidity, it really messes up its permeability properties. So there's a loss of permeability associated with fluidity. Could a membrane become too fluid, do you think? Yeah, it would just kind of lose integrity. It would kind of dissolve almost. What environmental factor might cause a membrane, if it wasn't controlled, to lose fluidity in that fashion, to dissolve, to become too fluid? Temperature, yeah. Increased temperature would increase motion of those phospholipid molecules. And so at some point, membranes could get too warm and basically dissolve. On the other hand, if membranes become too cold, then that loss of motion of the molecule could cause the membrane to become too static, and it almost freezes up. And that would have negative consequences as well. So a cell's got to be able to maintain membrane fluidity even though its external environment is changing. It's got to be homeostatic. So how can it do that? Well, take a look right here at two phospholipid bilayers. This particular phospholipid bilayer is made up of phospholipids, several of which have unsaturated fatty acids. Remember, fatty acids that are unsaturated have carbon-to-carbon double bonds in them? And when that happens, the hydrocarbon chain that's in the fatty acid has a bend in it, or a kink in it. So carbon-to-carbon double bonds form kinks or bends in the fatty acid chains. And they can't pack together as tightly within the membrane. So this would be a pretty fluid membrane. This particular membrane has primarily saturated hydrocarbon tails in its phospholipids. And those tails, because they don't have those bends in them, can pack more closely together. And therefore, this membrane would be more viscous. It wouldn't be as fluid. It would be more sticky. Okay? Cells can metabolically adapt and change what kind of phospholipids they're manufacturing to incorporate into their membranes. So, for example, at a particular temperature, this might be okay. A fairly viscous membrane might be okay. But if we start lowering the temperature even more, then this membrane can become too viscous and it could basically freeze up, lose its permeability. How could the organism respond? By adjusting its metabolism and start manufacturing more phospholipids that were unsaturated in nature and incorporate those into its membranes. Those wouldn't pack together as tightly, and therefore the membrane would maintain more fluidity even though the temperature was dropping. Plants can do this. Okay. Plants can't get up and move away if their environmental temperature starts dropping. And therefore they might be prone to their membranes freezing up. But they can respond metabolically. And they can start increasing the fluidity of their membrane to help compensate for the decreasing temperature by adjusting their metabolism and producing more of these unsaturated fatty acids in their phospholipids. The same could be said if temperature rises. If temperature rises, the membrane might become too fluid. And so how might you adapt? By increasing the production of saturated fatty acids, allowing the membrane to become a little bit more viscous, compensating for that increased temperature. Cholesterol also plays a role in maintaining fluidity. Cholesterol molecules serve as kind of a buffer. And they keep the membrane from getting too fluid when it gets hot, but too viscous when it gets cold. How do they keep things from getting too fluid? These cholesterol molecules hinder the movement of phospholipid molecules. It's like if you're trying to elbow your way through a room, and all of a sudden you ran into several really big people who just weren't going to let you pass. That would slow your rate of motion toward the door. Okay? On the other hand, these cholesterol molecules have this kinked structure. And like unsaturated hydrocarbons, they tend to maintain some of the fluidity. So they're kind of a buffer. Okay? There are several observations. There's a lot of data that has been collected over the years since 1975 that are in support of the fluid mosaic model. Let me just show you a couple of them. One is a technique, a microscopic technique called freeze fracture. In freeze fracture, tissue is exposed to a very rapid freezing environment, probably liquid nitrogen, probably under vacuum, and the cell just instantly freezes and becomes very brittle. Then if you section that tissue with something really sharp, like a diamond knife, that can actually split the cell membranes into the two halves. And so here's one of the phospholipid bilayers, and here is one of the cytoplasmic bilayers. And then you can, through electron microscopy, actually observe these particular membranes. One of the things that we notice when we do this freeze fracture is that there is an asymmetry. Remember what asymmetric means? Asymmetric means unequal. Okay? So there are differences in the environmental half of the membrane and the cytoplasmic half of the membrane. And that basically has to do with the protein composition. So some proteins tend to be associated with one side of the membrane. Some proteins tend to be associated with the other side of the membrane. Here you can see that the extracellular layer right here, that freeze fracture of that side of the membrane looks different from the cytoplasmic side. And so this is the evidence that really is supportive of this idea that the membrane is this mosaic. Okay? It's not just this two flat layers of protein sitting on the top. That there are proteins distributed all throughout the membrane in an unequal fashion. Another pretty cool experiment that somebody did, I'm not sure when this was done, involved taking cells from two different sources, mice and humans, and labeling proteins in the membrane with fluorescent antibodies. Okay, so the antibodies will attach specifically to the proteins. And the fluorescent antibodies glow a different color under fluorescent light. And you can see that under the microscope. And so they labeled mice cells with red fluorescent antibodies and human cells with blue. There are some tricks you can then do to these cells that causes them to fuse to one another and to form a hybrid cell. So they hybridized these mouse cells and these human cells. And then they saw what happened to the distribution of that fluorescent labeling. Okay? If the proteins were fixed within the membrane and didn't move, then half of the hybrid would be one color and half of the hybrid would be another color. But that's not what they observed. Instead, after time, they observed that the hybrid cell had a mixture of proteins from both the mouse and the human sources. So those proteins were moving throughout the phospholipid bilayer and weren't fixed in position. Again, more evidence in the favor of this idea of a fluid membrane. Proteins play a really important role in membrane structure and function. We've talked about the role that proteins can play in transport, both in active transport and facilitated diffusion. We also mentioned that proteins can have an enzymatic role. The sodium-potassium pump is both a transporter and an enzyme, an ATPase enzyme. Something that the second half of Chapter 5 talks about that we haven't had a chance to talk about yet is the phenomena of signal transduction. Signal transduction allows cells to respond to changes in their environment. And something that's going on outside of the cell can cause an effect in the cytoplasm of that cell. Signal transduction is really important in the functioning of hormones in your body. One important hormone in your body is insulin. And insulin plays a role in, obviously, regulation of blood sugar. What produces the hormone? What tissue in your body? Pancreas, right? And specifically, there are some cells in the pancreas called beta islet cells. Okay? So, in those particular cells, the genes for insulin production are being expressed, and that means the insulin protein is being produced, and it's being secreted by the beta islet cells and secreted into the blood. There are certain cells in your body that are targets for that insulin molecule. What makes them a target? They have a receptor. They have a binding site for that insulin molecule. Cells in your body that have a lot of receptors for insulin are liver cells and muscle cells. So when your pancreas starts cranking out insulin, it starts circulating through the blood, and it binds preferentially to these liver cells and muscle cells. And what does it stimulate them to do? To take up glucose, to lower that blood sugar level. Once they take it up, they store it in the form of a polymer, glycogen. So muscle cells and liver cells start accumulating glycogen in response to insulin stimulation. Well, insulin's a protein, so it's a pretty big molecule. And it's got polar amino acids. And the membrane isn't going to want to let that insulin molecule cross the bilayer. The insulin isn't permeable to the membrane. The membrane's not permeable to the insulin. So what happens? It binds to protein molecules in the membrane, and that binding basically sends a signal into the cytoplasm saying insulin has bound. And that signal activates various processes within the cell, causing it to take up glucose and then to convert that glucose into glycogen. So even though the hormone doesn't enter the cell, it can cause an effect within inside the cell. And that's the result of its interaction with some of these membrane proteins. One of the membrane proteins that's very involved in signal transduction is a class of membrane proteins called G proteins. Probably what's going to happen after the exam is one of the quizzes that I will give you will be kind of an optional quiz. It will be a chance to replace one of your other quizzes. And it will involve reading these few pages at the end of Chapter 5 about signal transduction in G proteins. I'm not sure we're going to have time to talk about those right now in class, although later on in the semester when we talk about molecular genetics and cancer, we'll come back and refer to some of these signal transducing proteins because they're important in some kind of cancers. We talked about the fact that proteins can be involved in intercellular joining, forming those gap junctions, for example, or those reinforcing desmosomes, or those tight junctions. We mentioned that membrane proteins can play a very important role in cell-cell recognition. One cell being able to determine if another cell belongs. And that's really a critical part of our whole immune system. Okay? So we've got immune cells constantly checking out cells in your body. Foreign, get rid of. Self, leave alone. So that whole idea of cell-cell recognition. So the proteins that are part of the cell membrane aren't just structural entities. They're not just kind of reinforcing structures. They really have a lot of varied function which is associated with the function of the membrane. So that's important to keep in mind. Okay, so that's a good place to wrap up in terms of exam material. So you will be responsible for your exam for things from the first day, which dealt with water. Remember that I said in Chapter 3 that I was not going to test you again on the macromolecules. There we go, thank goodness. But functional groups are something that you do need to be responsible for, and basic properties of carbon, hydrocarbon chains. Remember, you don't have to draw functional groups, but you have to be able to recognize them. So if I show you some candidates, you should be able to pick out an amino functional group, a carboxyl, aldehyde, ketone, phosphate, sulfhydryl. Chapter 4, cell structure. Know your cell organelles, obviously. Spent a lot of time talking about the endomembrane system in the context of cell organelles. Make sure you know what their functions are. And if we spent some time talking about how their structure was related to their function, that can be important. Probably the best example we gave of that was the mitochondria, how the structure was related to the function. And then the membrane stuff. Okay? You should be concentrating on your PowerPoint/notes and your study guide questions. I wouldn't spend time going back and rereading the textbook in its entirety. I would use the textbook at this point now if I had a question about something that was in my notes. And so using it more as a reference than you use it as a primary means of studying. And then the old exams. I think the old exams are good toward the end of your study, because that then can be a test about whether you're understanding some of the concepts or not. There might be things on the old exams that you don't recognize. I was looking through, and there probably are some study guide questions that you're not going to recognize, because I cover different things every year. And so one of the things you can ask tomorrow, if you have been looking through the study guide, is if you don't think a study guide question has been covered, ask me and I'll let you know. Okay. In terms of breakdown, since we've got a couple minutes, there will be 32 multiple choice. That is 64 points total, 2 points each. There will be 4 agree and disagree questions worth 4 points each. That's 16. And the remainder will be 2 10-point essays. Okay? Don't rush through the multiple choice, and yet at the same time, you've got to leave enough time to complete the exam. Don't leave questions blank. Okay? The essay questions are worded in such a way that everybody should be able to tell me something at least about the question. Okay? And the study guides give you a good kind of idea about what potential questions might be for both the agree and disagree and for the essays. Don't leave time on the table, okay? If you finish early, go back and review your exam. Now, you've got to be careful. Sometimes you'll go back and start changing answers you answered initially in the multiple choice. I wouldn't change an answer unless you had some good reason to do it. If you start second-guessing yourselves, more often than not, you change it from a right answer to a wrong answer. But sometimes, later on in the exam, answers are given away. Okay? A multiple choice question has 2 parts. It has the given information called the stem, and then it's got, for me, usually 5 choices. And stems usually contain factual information. And so sometimes the stem of one question can give away an answer in a subsequent question. Or it might give you some insight into an essay question. So going back and reviewing those particular questions and not walking out after 20 or 25 minutes is definitely in your best interest. Dr. Morton, any advice you want to give them in terms of exam?
Strongly recommend going over the previous tests that Dr. Browder has posted, if only to get a handle on how he approaches asking questions. It's not necessarily going to be the same as you had in 134. All right, so not only use those as a review of the information, but also the testing method. Those of you who were in 134 last semester, the first test of the first exam of the semester included biomolecules, and some of the stuff that has been covered here. That's a good place to go to, for some test questions. The study guide is probably your best bet. If you can answer the questions in the study guide completely, you won't have any problem with the exam. Now, downside of all this is that's a good bit of time. So you have to manage your time over the next several days to be able to have time to do this review. And a great way to know where your shortcomings are is to go over things and find out, I don't remember this. Go back, and only then go back to the textbook. Don't try to reread the textbook.
In terms of material within your notes, it's not enough to say, I recognize that. You really have to be able to explain it. And a good thing you can do when you're getting toward the end of your study, when you've already prepared yourself, is get together with somebody or a group and take turns explaining concepts to one another. If you can get up on a blackboard or a whiteboard and explain to someone how the sodium-potassium pump works, you should be able to handle any question I can give you. And so the real goal is not just to say, I recognize that. It is, can I explain it? Okay? And if you don't like to study with other people, do it out loud. Explain it out loud to yourself. If you do it in your head and just say it in your head, you will skip over the things that you have trouble with. But if you say it out loud, you'll find yourself stumbling when you're trying to explain if you don't understand. Or you can say one other thing.
Now, I've thoroughly reinforced that recommendation. Small groups of 3 or 4 people helping each other, asking questions, is a great way to study because somebody else will have a different perspective than you do. Now, if you're embarrassed because you think I'm not smart enough to be able to hold my own in a group, suck it up. It's a great way to learn.